Hello, everybody. My name is Winter Woodfield, and today, along with my partner Kendall Schmidt, we will be discussing the journal article entitled Nested Retrotransposition in the East Asian Mouse Genome Causes the Classical Non-Agouti Mutation by the author Tanave. And as you can see on the left side of the screen, there is an example of a wild-type agouti mouse.
So to give a little background, the agouti gene is the gene that's responsible for pigmentation in mice. In wild-type mice, which have the agouti gene, it secretes a paracrine signaling molecule, and that goes and tells melanocytes to make pheomelanin, which gives that kind of yellow or reddish-looking fur that you see with the mouse on the right. However, in mice with a mutation called non-agouti, that gene secretes a signaling molecule that tells those melanocytes to produce eumelanin, which gives the mice kind of a brown or black looking fur. So this gene is studied a lot in research and in mouse models because it's used to study the mechanisms of pigmentation and also how pigmentation can be linked to behavior and to epigenetic changes in the organism. This group went and looked specifically at the role of retroviruses as a cause of the non-agouti mutation, which was the mouse with the black-looking phenotype. And so to kind of talk a little bit about that, a retrovirus is an enveloped piece of RNA, and it replicates through reverse transcription. And specifically, when it becomes an endogenous retrovirus, this basically means that it's been incorporated into the germline so that it's integrated into an organism's genome and it can be passed on through generations. And so this functions as a class 1 transposable element. And so, as I said before, this entails that it moves around through the genome through an RNA intermediate, and then it's able to copy and paste into new areas. The reason that this group was looking into endogenous retroviruses as the cause of the non-agouti mutation was because there has been background research prior to this study, and that research found that an endogenous retrovirus called VL30 was inserted into an intron in the agouti gene. And up until this study was done, that was what was believed to cause that black phenotype. However, nobody really knew the mechanism in terms of exactly how it changed gene expression and at what point in time and from where that mutation and insertion got into the gene. So that was what prompted Tanave et al. To investigate this as their research question. Upon starting their research, Tanave et al. Discovered that there was an 11 kb insertion within that VL30 endogenous retrovirus. And that's the endogenous retrovirus that was in the agouti gene believed to cause the non-agouti mutation. And so they figured that given there wasn't any information about what that unknown insertion was within VL30, that might point to the cause of the mutation itself. So if you look in the figure, you can see right in the middle of that VL30 endogenous retrovirus in the mouse, there's a very sizable unknown element, and that was what they continued to pursue with the rest of their research. So the first part of investigating what this unknown element was to figure out where exactly did it come from, what kind of element was it. So the group used the mouse reference genome from a strain called B6 to kind of outline what the structure of that insertion was. So if you look in figure A, you can see that what they discovered first was that it has a 4 base pair target site duplication. And over at the right, you can see that that's just flanking the VL30 insertion. So this target site duplication is something that's characteristic of a retroviral insertion. So this was the first piece of evidence that told them that this unknown element was probably from a retrovirus. Then they went and saw that there was also direct sequence duplications from the VL30 element. If you go ahead and look in the second part of that figure, they had several of those sequences that were duplicated in various parts of the gene. And this is also something that proved to be evidence of a retroviral insertion in previous evolutionary time. So beyond these two pieces of information, they also saw that the sequence was almost 40% identical to envelope proteins that are in leukemia viruses, which was their third and kind of most critical information that this was definitely something that came from a retrovirus.
Next generation sequencing, NGS, was used to create a phylogenetic tree for the results. In the experiment, they analyzed the evolutionary relationships between the alleles of the agouti gene in inbred lab mice and wild mouse populations to determine where the non-agouti mutation occurred. The musculus group was sequenced using the NGS sequencing. As we have learned from class, NGS sequencing is a high-throughput sequencing method that works very quickly. Bioinformatic analyses piece together the fragments by mapping individual reads to that of the mouse genome. For this experiment, they sequenced the agouti gene locus. The phylogenetic tree was created based on the alignment of the full-length sequences of the agouti gene. When the phylogenetic tree was created, they discovered that the non-agouti laboratory mouse strains were clustered into the same group with the Japanese fancy mouse strain and other East Asian musculus group strains. NGS reads also revealed that most of the laboratory mouse strains with the non-agouti mutation have both VL30 and beta-4 insertions, but the wild mice only have a VL30 insertion in the agouti gene.
After doing next-generation sequencing, the group used NCBI BLAST and ended up finding that their unknown element was homologous to a sequence that was from an insertion in a piebald gene from an EDNRB gene, which is basically a gene that's from Japanese fancy mice. And so what this did was pave way to a possible origin for the insertion, because they knew kind of generally what area and region of mice that it came from and what kind of gene the insertion was from initially. So with this added evidence about the unknown element, they were able to compare this sequence to other exogenous and endogenous retroviruses in that figure. And that was essentially how they kind of narrowed it down. So with the phylogenetic comparison with those other retroviruses, they eventually figured out that the identity of this unknown element was called a beta-4 retrovirus, which is a type of endogenous retrovirus in mice.
The results showed that the two insertional mutation events happened in the agouti gene during the differentiation and domestication of the Japanese mouse. In the wild, the VL30 endogenous retrovirus was transposed into the wild-type agouti allele in the population of the musculus group. Then, the beta-4 endogenous retrovirus transposed afterwards into the VL30 sequence in the lineage that separates the Japanese fancy mouse from other East Asian wild mice. It is thought that the beta-4 insertional event has occurred relatively recently.
After making progress on figuring out a possible location of origin for the beta-4 insertion, the group decided to look at exactly how this insertion impacted the agouti gene expression in different mice. So to do all of their studies, they essentially looked at RNA expression data from two different types of mice. One of them was a CH3 mice, which is just a wild-type strain exhibiting typical agouti expression and phenotype. And the other one was a B6 strain of mice, which have the mutation causing the non-agouti gene and the eumelanin looking phenotype, which gives it that black, brown looking coat color. One key finding that the group noticed when they were looking at their mRNA data was that there were very different transcripts from the two different mice strains. So they used their mRNA, which was gauged using RT-PCR, and they went and compared the specific transcripts from the agouti and the non-agouti mice. So if you look at the figure, you'll see on the left, those B6 mice, which are the ones with the mutation, have an additional gene product, even though they have that insertion that's preventing them from expressing the agouti gene normally. Whereas if you look in the wild-type mice, you can see they do have that regular agouti gene product, as well as one additional transcript. And there at the bottom, they both have beta-actin, which was just used as a positive control. So basically what these results told them overall was that in the agouti gene for both the wild type and the mice with the mutation, they had very different overall splicing patterns. And that went and hinted at something else that could have been in play in causing that. The group looked further into seeing if maybe the beta-4 insertion inside of VL30 was this something that was causing the different splicing pattern and the different gene expression that was seen in agouti and non-agouti mice. So to see if this was the case, they deleted both the beta-4 and also the entirety of VL30 within the two different strains of mice that they were looking at before, the wild type and the non-agouti mice. So with this comparison, you can see that both of the mice exhibited normal phenotypes when they were in the control there on the far right. The mice with the VL30 deletions actually were able to have that regular phenotype rescued. So you can see that the B6 mice, which were the ones with the mutation, when they deleted VL30, they had normal expression. They had that normal red, yellow looking hair. So then one step further was to only delete the beta-4 insertion, which is inside of VL30. And when they did that, they saw that that in and of itself was enough to rescue the phenotype too. So that shows that it wasn't just VL30, but it was specifically the beta-4 insertion that caused that black-looking non-agouti phenotype. Because just by deleting beta-4, they could rescue it, even though those mice still had the VL30 insertion.
As we discussed earlier in our presentation, the agouti gene is responsible for the banded hair patterns of eumelanin and pheomelanin. If a large part of the beta-4 sequence is removed, then the non-agouti phenotype will change to the wild-type agouti phenotype. In this experiment, they used CRISPR to confirm that agouti coat color would be restored if the complete beta-4 sequence was deleted. However, they found that in the wild-type alleles, there was still a single long-terminal repeat of beta-4 in VL30, so they made the hypothesis that this long-terminal repeat was integral to the black and tan coat phenotype expression. A beta-4 allele called beta-4 direct recombination was created that still had the beta-4 long-terminal repeat, and the results showed mice that had yellow coat color on the ventral side but not on the dorsal hairs. To look further into this, they studied agouti transcripts expressed from 4 different promoters. In the figure of the gel, a real-time PCR was performed of the agouti transcripts in the dorsal and ventral skins of the beta-4 direct recombination and the beta-4 deletion mice. The beta-actin was used as the control in this gel because every individual mouse has it. At the top, the D and V stands for dorsal and ventral respectively. The results showed that the black and tan phenotype is caused by a single beta-4 long-terminal repeat sequence that completely disrupts the expression of the agouti transcript from the 1B, 1C in both dorsal and ventral skins, but it does not disrupt the expression of the agouti transcript from the exons 1A, 1A' in the ventral skin. This is because different promoters are governed by different parts of the body, meaning that there are dorsal and ventral promoters. The splicing patterns are the cause of the different exon start sites, and that is what will result in the dorsal and ventral patterning. Since this article was published, the author has released another article discussing the role of beta-4 in the mouse genome, specifically in the development of Japanese fancy mice, which you can see an example of in the picture to the right. In the future, experiments could be conducted using epigenetic aspects to figure out the details of the mechanisms that are involved in retroviral elements. In this journal article, they found that the black and tan phenotype is caused by the solo long-terminal repeats of the beta-4 and the agouti gene, but it could not be determined to exclude the significance of the VL30 sequence in altering the expression of the agouti gene. Further experiments could be done to figure out if VL30 does in fact alter agouti expression.